Hey guys, welcome back to Joanne's Learning Hub. In today's video, I'll be explaining cell structure of A-level biology. This is a mind map of the syllabus of cell structure, which you can also see on the Cambridge site. Now, cell structure is divided into two topics: microscope in cell studies and cells. Now, the first two topics, slide preparation, drawing cells, eyepiece gratitule and stage micrometer, these are more towards the practical side. So I'll be beginning with magnification and the difference between resolution and magnification. There are two main types of microscopes, light microscope and electron microscope. Now, what's the difference between the two? Light, as the name suggests, uses its source of radiation as light, whereas for electron microscope, the source of radiation is electrons. The resolution of light microscope is 200 nanometers, whereas for electron microscope it is 0.5 nanometers. The lens used for light microscope is glass, whereas the lens used for electron microscope is an electromagnet. When we use a light microscope, the specimen is alive and the image formed is a colored photomicrograph. On the other hand, for electron microscope, the specimen is dead and the image which is formed is black and white. Electron micrograph, it's called. When we use a light microscope, it's viewed using the eyepiece, whereas for electron microscope, it is viewed using a fluorescent screen. And light microscope uses stains as colored dyes, whereas for electron microscope, the stains is heavy metal. One thing you need to remember about electron microscope is that vacuum is present. Why is the vacuum present? To prevent electrons colliding with air particles. Now, even in electron microscope, there are two types. One is transmission electron microscope and the other is scanning electron microscope. The difference is in the resolution mainly. For transmission electron microscope, the resolution is 0.5 nanometers, whereas for scanning EM, the resolution is 3 nanometers. Now coming to resolution and magnification. What is the difference? Magnification, a topic which we've even learnt in our IGCSE biology, it is the number of times larger an image of object is than real size. So it has a formula. Magnification is equal to the size of image divided by the real size. Resolution, on the other hand, is the ability to distinguish between two separate points. So as the resolution increases, image clarity and detail also increases. For light microscope, the magnification comes up to 2000. Whereas for electron microscope, the magnification is over 500,000. Now, light microscope has some advantages over electron microscope. One is that since the specimen is alive, you can observe living tissue. It's more portable. This is because electron microscopes are large and installation means it cannot be moved easily, which makes light microscopes more portable. So they are also easy to use. You can see real or natural colors and the live specimen. When you use light microscope, you can stain particular types of tissue for better visibility. Another thing I would like to add is that light microscope has two types of lenses. One is an eyepiece lens. It usually has a magnification of 10, and then it also has objective lens, which is a series of 3 objective lenses with different magnifications. So now this comes in handy when we are calculating magnification. There's a formula which is there: eyepiece lens magnification plus objective lens magnification is equal to the total magnification. So this is what gives us the magnification of the specimen. We may also be given the image size or the actual size, and then we can find out the other value. So if we have the image size, we can find the actual size, and if we have the actual size, we can find the image size. Moving on to cells. Now, cells are the basic units of all living organisms. In the topic of cells, we'll be learning about eukaryotic cells and prokaryotic cells and also the difference between the two. We'll also be covering the role of ATP and viruses. Now, in eukaryotic cells, we need to know the structures and functions of these 15 organelles or structures which are present in the eukaryotic cells. This includes cell surface membrane, which is basically the cell membrane, the nucleus, the rough endoplasmic reticulum as well as the smooth endoplasmic reticulum, the Golgi body, mitochondria, ribosomes, lysosomes. Lysosomes are the societal bags of the cell. Centrioles and microtubules, cilia, microvilli, chloroplasts, cell wall, plasmodesmata, and the large permanent vacuole and the tonoplast of plant cells. Now, before we begin discussing about cell surface membrane, you need to know these three definitions of organelle, eukaryote, and prokaryote. Now, an organelle is a functionally and structurally distinct part of a cell. Eukaryote is an organism whose cells contain a nucleus, 1, and 2 membrane-bound organelles. So then what's prokaryote? Prokaryote is an organism whose cells do not contain a nucleus or membrane-bound organelles. Now, animals and plant cells, they come under eukaryotic cells. Cell surface membrane, they are present in both plant and animal cells. So, this cell membrane is something we've even learnt in our IGCSE biology. Most of these structures we've learnt there. Cell membrane, it is thin, it's partially permeable, and it allows for the exchange of biological molecules and ions. It's a barrier between the cytoplasm and the external environment, and it helps with cell recognition. Now, one thing you must remember about cell membrane is that it's formed from a phospholipid bilayer of phospholipids. And phospholipids is a topic we'll be covering in chapter 2 of A-level biology. The nucleus is present in all eukaryotic cells. It is relatively large and it's surrounded by a double membrane nuclear envelope. The nucleus is continuous with the rough endoplasmic reticulum. The nucleus contains nuclear pore, nucleolus as well as chromatin. So what are these three? The nuclear pore is basically gaps in the nuclear envelope. Remember that the nuclear envelope is a double membrane around the nucleus. So the nuclear pore is the gaps in the nuclear envelope, and this allows exchange between nucleus and cytoplasm. The nucleolus contains loops of DNA from several chromosomes. So it basically synthesizes or manufactures ribosomes. That's the function of the nucleolus. Nucleus also contains chromatin, which is the material from which chromosomes are made. So the key points under nucleus are, one is that it's a large dense organelle. It has nuclear envelope surrounding it, and this nuclear envelope is double membrane. The nucleus is continuous with the RER. Nucleus contains nuclear pore, which is basically gaps in the nuclear envelope. This is allowing exchange between nucleus and cytoplasm. The nucleolus, it contains loops of DNA and it synthesizes or manufactures ribosomes. And the chromatin is the material from which chromosomes are made. Mitochondria is the powerhouse of the cell. Its function is to carry out aerobic respiration, and it also synthesizes ATP. The full form of ATP is adenosine triphosphate. You must note that mitochondria is only visible with a light microscope, not with electron microscope. Since mitochondria is the powerhouse of the cell, it's present in greater number in cells that require more amount of energy. The structure of mitochondria goes like this. Its outer membrane contains a transport protein called porin, and its inner membrane is folded to form cristae. Now, what is the reason behind the inner membrane being folded? It's to increase the surface area. The rough endoplasmic reticulum, or RER. Why is it called rough? Because its surface is covered in ribosomes. Now, these ribosomes are the ATS ribosomes, which are the sites of protein synthesis. The RER is a single membrane organelle, and it forms an extended system of fluid-filled sacs, and these sacs are called cisternae. The proteins made by these ATS ribosomes enter the sacs and then move through them. Smaller sacs, also called vesicles, they break off from the endoplasmic reticulum and they join together to form something called the Golgi body. Smooth endoplasmic reticulum, as the name suggests, it's smooth, which means that it does not have ribosomes present on its surface like the RER. The smooth endoplasmic reticulum is involved in the production, processing and storage of lipids, carbohydrates and steroids, which includes cholesterol and reproductive hormones, for example. Golgi body, which is also called Golgi Apparatus or the Golgi complex, it's a stack of flattened sacs of membrane which is similar to the smooth endoplasmic reticulum. So the flattened sacs is called cisternae, and it's formed by the vesicles that, as I told you earlier, bud off from the rough endoplasmic reticulum. The Golgi body is a single membrane organelle. Its main function is to modify proteins and package them into vesicles or lysosomes. It also synthesizes glycoproteins and glycolipids. Now, the word glyco suggests that it's a carbohydrate group, protein. So, carbohydrate group plus protein forms glycoproteins, and carbohydrate group plus lipids form glycolipids. During plant cell division, Golgi enzymes are involved in the synthesis of new cell walls. Now, this is a rough diagram which includes the nucleus, the nucleolus, the nucleopore, the nuclear envelope, the Golgi body, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. You can also see the chromatin and the nucleoplasm. Lysosomes are the suicidal bags of the cell. How did lysosomes get this name of suicidal bags? This is because they are specialist forms of vesicles which contain hydrolytic enzymes. And what are hydrolytic enzymes? They are enzymes that break down biological molecules. Now, lysosomes are responsible for the digestion or breakdown of unwanted structures or waste materials such as worn out organelles. So, it's used extensively by cells of immune system, and lysosomes also perform endocytosis, exocytosis and self-digestion. Ribosomes are formed in the nucleolus, and they are composed of almost equal amounts of RNA and protein. Ribosomes are found freely in the cytoplasm of all cells or as a part of the RER in eukaryotic cells. Now, ribosomes are composed of two subunits, small subunit and large subunit. So, we know that eukaryotic ribosomes have ATS ribosomes. So, this ATS is divided as 60S and 40S subunits. And eukaryotic ribosomes are found in the RER and the cytoplasm. Prokaryotic ribosomes contain 70S ribosomes, and the 70S is divided into 50S and 30S subunits. Prokaryotic ribosomes are found in the mitochondria and chloroplasts. Ribosomes carry out protein synthesis of the cell. Now, cell wall is a cell structure which we've learned from childhood. It is fully permeable It prevents the cell from bursting. This is something we learnt in our IGCSEs, so it allows turgidity. The cell wall is rigid as it contains fibers made of cellulose, and the cell wall gives a definite shape to the cell. Microtubules. These are long, rigid and hollow tubes which are found in the cytoplasm, and they are made of a protein called tubulin. Microtubules makes up the cytoskeleton of the cell. Now tubulin has two forms, alpha tubulin and beta tubulin. Alpha and beta tubulin molecules combine to form dimers, and several dimers joined together end to end form long protofilaments. 13 of these protofilaments line in a ring to form a cylindrical structure with a hollow center, and this is called a microtubule. So this is a flowchart from tubulin, which is alpha and beta tubulin, which forms dimers. Then dimers are joined end to end to form protofilaments. 13 of these protofilaments will form a microtubule. And you must remember that microtubule has a hollow center. Now, since microtubules make up the cytoskeleton of the cell, it determines the cell shape and it also supports the cell. The assembly of the microtubules from the tubulin is controlled by special locations in cells, and this special location is called the microtubule organizing center, otherwise known as MTOCs. Centrioles are formed by 9 triplets of microtubules. Centrioles are hollow fibers which are made up of microtubules, and when two centrioles join at right angles to each other, they form a centrosome. So centrosome is made up of centrioles, just like how centrioles is made up of microtubules. Now centrioles produces the spindle fibers and the centrosome organizes the spindle fibers during cell division. You must remember that centrioles are not found in flowering plants and fungi. Cilia is hair-like projections which are again made from microtubules. They allow the movement of substances over the cell surface. Microvilli is cell membrane projections and they help to increase the surface area for absorption. Chloroplasts, they are structures found in plant cells and they are found in the green parts of a plant. We say that chloroplast contains the green pigment chlorophyll. Note that chloroplasts, they are larger than mitochondria, and chloroplasts also are surrounded by a double membrane. So if we observe the structure of chloroplast, it has an outer membrane and inner membrane. The space between the outer membrane and inner membrane is called the intermembrane space. Now, membrane bound compartments is called thylakoids. It's only one of the discs, as you can see. When these thylakoids stack up together, a stack of thylakoids is called the granum. Now, if we see carefully, the granum, all the stacks, they are joined together by something called the lamella. So, the lamella is the thin and flat thylakoid membranes which joins the grana. We know that chloroplasts, they are the sites of photosynthesis. The light-dependent stage of photosynthesis happens in the thylakoids, whereas the light-independent stage of photosynthesis happens in stroma. And now stroma is an aqueous fluid. Chloroplasts also contain starch granules, circular DNA, and 70S ribosomes. ATP is also produced in chloroplasts. Now, Plasmodesmata is a new structure that you would be learning. They are the pole-like structures which are found in the cell wall of cells. The role of Plasmodesmata is to allow a link between neighboring cells by fine threads or cytoplasm. It allows the transport of water, sucrose, amino acids, ions, etc. between cells without crossing membranes. And this kind of movement is called a symplastic pathway. The Plasmodesmata also allows communication or signaling between cells. Vacuoles are sacs surrounded by a partially permeable tonoplast. And now the tonoplast is a selectively permeable membrane which controls the exchange between the vacuole and the cytoplasm. Vacuoles help regulate the osmotic properties of cells. The fluid present in vacuoles consists of plasmog. Now this is a mnemonic which you can use to remember what is present in the fluid of vacuoles. So P stands for pigments, E stands for enzymes, S stands for starch, O for organic molecules, M for mineral salts, O for oxygen, and C for carbon dioxide. So you can see the structure of a large permanent vacuole. The outer membrane is called the tonoplast. The inner membrane is called the cell sap, and then the small structure right here is the central vacuole. Now you must remember that the vacuoles present in animal cells are not permanent and they are small, whereas in plant cells the vacuoles are permanent and they are large. So this is a large permanent vacuole. ATP plays a vital role in all organisms. This is because all organisms need a constant supply of energy to maintain their cells as well as stay alive. This is why ATP is known as the universal energy currency. You must remember that ATP is a nucleotide and it's required in processes like anabolic reactions, which is basically building larger molecules from smaller molecules, to move substances across cell membrane and also within the cell. And specifically in animals, the energy provided by ATP is used for muscle contraction and conduction of nerve impulse. Now let's discuss about prokaryotic cells. As I had said before, prokaryotic cells do not contain nucleus, nor do they contain membrane-bound organelles. Structural features of prokaryotic cells include that they are unicellular. They are generally between 1 micrometer to 5 micrometers in diameter. They have circular DNA, but they do not have endoplasmic reticulum. They have 70 S ribosomes. Of course, there is no membrane around organelles, which is why it's prokaryotic. Its cell wall is made up of murine and also peptidoglycan. And peptidoglycan is made up of polysaccharide and protein or amino acids. So polysaccharides plus amino acids. It lacks nuclei or proper nuclear membranes as well. Now you must also know that there are some structures which are always present in prokaryotic cells, especially bacteria, which is an example of a prokaryotic cell. You must remember animals and plants are example of eukaryotic cells. Cell wall, cell surface membrane or the cell membrane, cytoplasm, circular DNA and ribosomes. These structures are always present in the prokaryotic cells. Structures which are only sometimes present includes capsules, flagellum, plasmid, which is a small circle of DNA, pili, infolding of cell surface membrane. Viruses is something we've even learnt in our IGCSE biology. They are non-cellular infectious particles. They are parasitic in nature as they reproduce by infecting and taking over living cells. The structure of viruses includes a nucleic acid core. Their genomes are either DNA or RNA, and this can be single-stranded or double-stranded. It also contains a protein coat which is called a capsid. Some viruses have an outer layer called an envelope, formed usually from the membrane phospholipids of the cell they were made in. This is a sample question of magnification. So you have been asked the actual diameter of the nucleus, you've been given the magnification. if you have the physical copy of the paper, you're supposed to use your scale to measure the diameter of the nucleus, so that becomes the image size. Since here all the actual diameters have been given in micrometers, you must do a conversion. 1 mm is equal to 1000 micrometers. And using that conversion, you can apply the formula magnification is equal to size in image upon real size, or the actual size. And since you need to find actual diameter, you rearrange the equation to make it actual size or actual diameter is equal to size in image divided by the magnification.